So Sophus Lee developed this particular method of basically applying the ideas that were developed by Évariste Galois, who used these ideas of groups to explain why certain polynomial equations could be solved and why other polynomial equations could not be solved. So Lee noticed that a number of different equations that were solved by ad hoc techniques admitted a group of transformations to make the equation invariant, and he used this idea to unify the different methods and indeed to solve equations that couldn't be solved. A lot of the work that was done in the 20th century applied the method of Lee in mathematical physics. In addition to his work, I also mentioned the work of Emmy Noether. He applied Lee's ideas, well, the ideas of invariance applied to action integrals, and used those ideas to actually derive a formula that not only found the so-called Noether symmetries, but also gave a direct formula for the calculation of first integrals or constants of motion. This is, of course, very popular with the physicists because without thinking, they could just substitute into a formula. They didn't have to do any differentiation or, heaven forbid, integration to work out these constants of motion. Emmy Noether is an interesting fact. We often talk about Lee, but we don't talk too much about Emmy Noether. He's well known in the pure community. Very few people know of his work in applied mathematics. Indeed, as a typical pure mathematician, Emmy Noether didn't like the work she did in applied mathematics. She literally called it crap and didn't want anything to do with it later in her life. It was a typical pure mathematician who didn't want to do anything applied at all. But of course, for those of us who have used a method, we use it very, very successfully to solve a number of different problems in mathematical physics. And I'll say a little bit about the method later and shows a little application of that with one particular problem as well. Okay, so what is the method? How do we apply the method? Well, basically, we're looking for, we're analyzing a particular differential equation. We say that the equation possesses the symmetry of this form here. This is a linear operator, which operates on the equation when we extend the symmetry to the same order as the order of the equation. And so here we have the n, which we call the nth extension of G. There's a standard formula for working out the nth extension. I won't go into the details of that. Various software packages have been written to be able to solve the horrible equation that arises when we operate on the equation with the nth extension of G. Okay? We say that G is the symmetry of the equation, or the generator of the infinitesimal transformation that leaves the equation invariant. If you have the infinitesimal transformation, it's rather easy to obtain the global form or the finite form of transformation. You just have to solve this system of equations. If you solve this system of equations subject to these initial conditions, then we'll have x1 is a function of x, y, and epsilon, y1 is a function of x, y, and epsilon, and rather interestingly, this x1 and x2 then form a group. And this is what was so remarkable about these particular results. Just by looking at invariance properties of equations, you now have this rich structure of group theory underlying everything that you can do. So once you find the symmetries, the question is, well, what do you do beyond just providing the finite form of the transformation that leaves the equations invariant? Well, obviously, you want to solve the equations. So for ODEs, what we tend to do is we first look at reduction of order. So what is very nice about the Lie symmetry approach is that once you have a symmetry, you have a systematic way of reducing the order of the equation. Importantly, you find your reduction variables by solving what we call an associated Lagrange system or characteristic system. This will give us two invariants, one that just depends on the variables, the second one that depends on the variables and the first derivative. And once you use these two variables, you can write your original equation of order n now as an equation of order n minus 1 exclusively in these new variables. This is the power of the method of Lie. Once you define these variables, the reduced equation will only be in terms of these new variables. It's surprising that even for a number of us have been using these methods for many, many years, there's many practitioners who don't use this very systematic way of reducing the order of equations, and you will still find papers being published today where people try various ad hoc techniques, ad hoc relationships between the variables in order to reduce the order of the equation. Invariably, they get a simplification. Simplification is indeed what you'd obtain when you actually use Lie symmetries in the first place. Another nice property about the Lie symmetries is that they form a Lie algebra, okay, and as a result, one can reduce the order of quadratures depending on the structure and dimension of the algebra that one obtains. In fact, Lie proved the result for every Lie group you have a Lie algebra, and associated with every Lie algebra is a Lie group as well. So you have the Lie algebra associated with symmetries, and the Lie group associated with the finite transformation itself. You can also use symmetries to specify equations. You can determine equivalence class of equations via these Lie algebraic representations, taking this Lie algebra into account. For example, if you're looking at second-order ODEs, we know that they possess 0, 1, 2, 3, or 8 symmetries. You have Lie algebras associated with all these symmetries, but 0 and 1 case. For all the others, you have more than one Lie algebra. The very interesting case, that of 8 Lie point symmetry, which we know is the case of linear or linearizable equations. And so if an ODE, a second-order ODE, admits 8 Lie point symmetries, even though it may not be linear, you can certainly linearize it. And very nicely, not only can you linearize it, but you can actually linearize it to y double prime equals 0, the three-particle equation. Okay, so that's some rather obvious applications of these symmetries to ordinary differential equations. You can also use symmetries to determine first integrals or constants of the motion or conservation laws. You can certainly do this using these symmetries, but the calculations, especially for ODEs, become incredibly technical. The quasi-linear equations are often intractable, and so it is quite difficult to actually use these symmetries. However, what is very nice is that there's, as I mentioned earlier, direct calculation of first integrals using Noether symmetries. You'll need an action integral though. You don't just look at the equations. I'll say a little bit more about that slide. An area of applications of symmetries that is perhaps ignored and not fully appreciated is the idea of group invariant solutions, particularly for ODEs. What is nice about this particular approach is that it obtains singular solutions for ODEs. So in addition to reducing the order of the equations using symmetries and obtaining the general solution, or at least reducing the ODE to quadrature, you can also obtain singular solutions of ODEs, provided you can solve this particular equation here, y prime is eta over psi. You substitute this into the equation, and then often you get a solution, so you get 0 as a solution, and then you discard that. Okay. This is a new area of research. I'm starting to do a little bit more work on solutions of ODEs, but I won't go into that in detail today. Perhaps the best known application of group invariant solutions is for PDEs. You can use symmetry to reduce the number of variables of the PDE. You do this to reducing it by 1 in each case. Ultimately, you want to reduce your PDE to an ODE, and hopefully you can solve that. Okay. 2 points that I want to make in this regard. Firstly, once again, if you use Lie symmetries, you can always write or your reduced equation in the new variable. You won't have any of the old variables around at all, so this gives you a systematic approach to reducing the PDE. The downside, though, is that when you find these group invariant solutions, these are not going to be the general solutions of your PDEs. Okay, these are just going to be particular solutions of your PDE. However, what we've found in certainly in applications in mathematics and physics is that these solutions tend to be the physically important solutions. And we hope that these are also going to be the biologically important solutions when we apply these methods in mathematical biology. Okay. Interestingly, the fundamental solution of the heat equation can also be obtained via group invariant. Okay. Looking at examples, I just want to say a little bit about Noether symmetries. As I said before, the Noether symmetry approach requires an action integral, and it in fact requires the invariance of the action integral under a one-parameter family of transformations. If we just consider first-order Lagrangian, okay, then the condition for a Noether symmetry is given by this very simple equation here. And so it's not too difficult to solve that equation. As I said earlier, regardless of the symmetry, you can always write down first integral. So for every new dissymmetry, it's associated a first integral of this particular form here, given by equation 9. This is also why we tend to say that when one has a new dissymmetry for a particular system, one is allowed to actually reduce the equation twice, because the first reduction gives you the first integral, then you keep the first integral as an equation which admits this particular new dissymmetry, and you can reduce the order. Okay. So I'm going to use Lie symmetries and the odd-case new dissymmetries to attempt to solve some equations that are written in various applications in mathematical biology. The first example I want to present to you is a very, very simple example. It's the so-called SIR model. I think it's a special case of the famous Kermit and Kendrick models. In this SIR model, you have three compartments. The first compartment, S, contains susceptible individuals. Your second compartment, I, is the compartment of infected and infectious individuals. Okay? And the final compartment, R, is those that have been removed from the infectious compartment, so they're no longer infectious. They've been removed either because they're now immune to the disease, covered and they're immune, or they have, so in some sense, they've been removed from the system. So when we set these models from this particular and admittedly very simple system, we have that the concentration of individuals in the susceptible population will be reduced as a result of contact between those individuals and infectious individuals at some contact rate beta. Okay? So the number of individuals in the first compartment will reduce. Okay? In the second compartment, with the infectious individuals, the concentration of individuals will increase, again due to this contact rate, beta. And in addition, the number of individuals in the compartment will reduce due to some sort of recovery. Okay, and we'll call that rate alpha. And finally, the number of individuals in the recovered compartment will increase as a result of this recovery rate. Okay? So these are the three first-order OBEs governing the behavior of this particular system for a particular individual. It turns out, of course, that we don't look at the entire system. Since R doesn't appear in the first two equations, it actually doesn't affect the dynamics of the disease at all, and so we can ignore the third equation, and we just consider these two equations. So this is a very simple system. It is nonlinear, okay? And so usually what one does in biology is you undertake a dynamical systems analysis of this particular system. Now, if you do this, the challenge that we have is that the equilibrium points are weak, they're not robust, they're actually at the interface of stable and unstable regions, and so you cannot extrapolate from the linear regime to the fully nonlinear regime in which this particular system lives. And so you can't really make conclusive statements about this particular problem when one looks at a dynamic. And so what we tend to do is look at the system from a Lie symmetry perspective. Okay? But of course, for those of us who work in symmetries, we know that you cannot find Lie symmetries of first-order equations directly. Okay? And this is indeed the case in this particular system. And so what we need to do is combine the system of two equations into a simple equation. So if we solve the very first equation for I, we obtain apologies, we just seem to have a technical issue with the computer. Okay, my apologies. My screen seemed to have completely frozen with regards to that finally seems to be. Right, okay, thank you. Apologies for that. Right, so you solve the first equation for I, and you obtain I as minus S prime over beta S. You substitute into the second equation, and you get this nonlinear second order. Okay. Unfortunately, when you analyze this equation for symmetries, regardless of the values of alpha and beta, you only obtain the single symmetry partial T. Now, if you have a second order equation with one symmetry, that usually means the best you can do is reduce the order by 1. Okay? And once you've reduced the order by 1, that is all you can do. Okay? However, this equation can actually be integrated, which suggests there's a little bit more going on. Indeed, if we analyze this equation a little bit more, we write down a Lagrangian for this. It's a rather curious-looking Lagrangian with all these logs running around. Excuse me. But nonetheless, it is a Lagrangian, and the symmetry associated with this Lagrangian is indeed this same symmetry, G1 here. Okay? So G1 is not just a Lie symmetry of the equation, it is a nude symmetry associated with this particular Lagrangian. And since we have a nude symmetry, then we can go ahead and write the first integral given here using the formula that we've used. And so even in this very simple system where you only have a single symmetry, you can certainly reduce the order and write the equation down as a quadrature. Okay. So it's going to be fairly easy to write. And just to distinguish between this equation and the Lagrangian, I've switched from S's to Y's. The S's and the Y's are exactly the same. There isn't a transformation. But mathematicians prefer to use X's and Y's. We don't like using S's and I's, which tend to confuse those sorts. Okay. So, given that one has this particular solution for that simple model, the question is, can you do this with more complicated models? And if you've attended my talks before, have seen this particular example before, so I'll go over it very quickly. So again, we have this so-called bilinear or quadratic system. It's a two-strain system, two classes of infections, I1 and I2. And you can see that the concentration of individuals in the I1 class grows at a particular rate. Okay? It decreases as a result of interactions within the population, and it also has interactions between the two classes, I1 and I2. I don't want you to worry too much about the biology of this particular problem. All I want you to understand is this is a fairly standard set of equations that one looks at when one deals with infectious diseases. So this is slightly more complicated than the system that we looked at previously. If you do a dynamical systems analysis here, you'd find that you have 4 equilibria, and you can do an analysis for the 4 equilibria. When it comes to stability, we have 18 consistent robust cases and 34 consistent marginal cases. And of these different cases, you have 7 unique marginal cases. And by marginal, as I said earlier, what this means is in these particular cases, you cannot extrapolate from the linearized system back to the fully nonlinear system. Okay. And as a result, you have to make other techniques. In our case, we undertake a symmetry analysis of the system. So we solve the first equation for I1, substitute it into the second equation, and then obtain a single equation. I've used the variable u. This variable is just your I2. Okay. And as you can see, even from the simple quadratic system, we get a fairly horrible-looking equation that we need to analyze. In spite of how nasty the equation looks, you can still make progress. Okay? And just to give you one example, in a particular case, when we have the beta 2 equals beta 1 minus mu, the equation is 8 Lie point symmetries, so it's a linearizable equation. You can say a little bit about the biology of the equation. R0, your basic reduction number, is greater than 1, which means that you have an epidemic. Your the endemic equilibrium for E1 is locally asymptotically stable, so the solution goes to that particular equilibrium point, while the disease-free equilibrium is unstable, and therefore you understand that you have an epidemic. Okay? So you can see a lot about the biology as a result of looking at the symmetry analysis of the particular system. So what is really interesting is that while the symmetry analysis has all 7 of these cases, using center manifold theory, which tends to be a fairly powerful method, one can only handle 3 of the cases that arise here. So again, showing that symmetry analysis is a case where other methods fail in the analysis. Okay. So both the systems are presented to you with with just 2 dimensions. The question is, can we do this in high? But of course, once you move to high dimensions, it becomes far more complicated. So if you look at the first system I've put up here, okay, trying to write this system of 3 equations as a single third-order equation is indeed quite difficult, if not impossible. Okay? the least restrictive simplification in order to make the system tractable is to ensure that A13 here is 0. And so you can write the system in this particular form here. Okay. In spite of making that simplification, it's still quite complicated to look at, and one of my students is looking at this at the moment, and the best he's been able to do is write it down as this horrible-looking third-order equation. We have a number of constants running. A's are the same A's presented here just now. We also have some constant K's running. If you want a list of those constants, it runs into 8 or 9 pages, so it is really quite a mess. And given that it's quite a mess, I will do all the analysis. I hopefully we'll be able to present that at some future stage. Okay. So that was just to tell you briefly about applications of symmetry methods to ordinary differential equations that arise in mathematical biology. You can also apply it to some systems of PDEs that arise in mathematical biology. And I just want to show you how you can build up a very simple PDE model in MathBio. So we're all aware of so-called Malthusian growth, exponential growth, where U dot is just R U, where R is your growth rate. If you solve that equation, you have exponential growth, where growth just continues unboundedly. It's not very feasible, but for a short time, in lab situations, one can use Malthusian growth to estimate the growth of particular populations. A more realistic growth is the so-called Verhulst equation, where we have logistic growth. So here you have a new parameter k that is introduced into the equation. This is called the carrying capacity, and as a result of this form of the equation, your population is not allowed to grow beyond that carrying capacity. Okay. And so this is far more realistic when one looks at population growth. If you want to extend these ODEs to PDEs, so we now need to include spatial movement. We tend to include the spatial movement via diffusion. And so we now have this equation down here. We have ut is dUxx. So this is the diffusion. D is your diffusion constant. And here is your logistic growth. This is the celebrated Fisher's equation, which was initially introduced to model the propagation from gene. It also arrives in propagation, neurophysiology, and a number of applications in physics and in mathematical biology. Interestingly, it's recently been used to model sexy fly movement, and I have a student looking at this particular equation and matching it to data of the movement of sexy flies in KZN. Okay. And I should just point out this is Fisher's equation. It's not the Fisher equation which arises in finance, that is something completely different, and is not a PDE at all. So if we look at the symmetries of Fisher's equation, you'll notice that I've rescaled the constants d, r, and k out of the equation. You can always do that. If you do a symmetry analysis of this equation, that unfortunately only has two symmetries, the symmetries that you could have assumed just by looking at the equation. It's very easy to plot these particular symmetries. If you do the reductions by each of these symmetries, reduction by G1 just leads to the standard Verhulst logistic equation, which is what you expect because you're removing the spatial dependence again. If you look at reductions by G2, this just leads to the steady state or the static solution of the system. Equation 21 is a simple equation to solve, and it's easy to write down the solution. The more interesting solution to find is not really the reductions by G1 and G2, but it's a reduction by a combination of G1 and G2, and it leads to the so-called traveling wave solution. And this is also something quite interesting, that a number of practitioners who try to look for traveling wave solutions just make this particular answer down here. They let W of Z equals W of X minus CT, so the traveling wave answer is Z equals X minus CT. But of course, it has a group theoretic basis. Okay? It's a result of the equation being variant under partial T and partial X separately. So if we now substitute W of Z equals W of X minus CT into the original PDE, you obtain an ODE, which is slightly more complicated than the previous ODE. And it turns out that this, unfortunately, just has a single symmetry, which is, sorry, my apologies, this Wx down here should be a Wz. So this is just an equation that's W as a function of Z, and it has just a single symmetry, Wz, and this is not a new symmetry, so you cannot actually solve this equation symmetry. However, if you do some further analysis of the equation, you find that it has an additional symmetry when C is 5 over the root of 6. Okay, so in that particular case, you have a second symmetry. You have a second order equation with 2 symmetries, and we can now reduce it to quantity. And this particular value of C was also obtained by Pavlovitz and his collaborators when they undertook a Penlevé analysis of this equation. Okay? So they again underscored the things that a number of us have reported over the years between Penlevé analysis, which is a singularity analysis, and symmetry analysis, which is. Curiously, if you undertake a dynamical systems analysis of this particular equation, the value of C given for symmetry is not relevant at all. In fact, it's not significant at all to the long-term behavior of the system. In fact, the only value of C that is important is C equals 0. So C less than 0 has particular stability for your fixed points. C greater than 0 has a different stability for one of the 3. Okay? But certainly not the values of C reported from the symmetry analysis. So this was the Fisher's equation in one dimension. Obviously, one would like to look at it in higher dimensions as well, because we don't tend to just have one-dimensional movement. And the obvious generalization is to 2 dimensions. And interestingly, as soon as you do that, even though you're just looking at movement in 2 dimensions, this immediately imposes rotational symmetry on the problem. That is evidenced by looking at the symmetries of this particular problem. The G1 and G2 is what you had before. G3 is because you're introducing a new dependent variable implicit problem. Sorry, independent variable implicit in this problem. And this additional symmetry here is the generator of rotations in the plane. And so this is where you have your rotational symmetry. And I have one of my honor students doing a little bit of analysis of this particular system and writing it down as a dynamical system and trying to understand exactly what is going on in this particular system here. Okay. So that was a fairly simple PDE that we've analyzed. One could look at slightly more complicated PDEs. Let me see if this is going to work. I hope it does. Right, okay, so I just want to show you a little video here. So that is, so the next application that I want to look at is application in chemotaxis. And so chemotaxis is the orientation or movement of an organism in response to a chemical signal. Okay? And what you're looking at here is the movement of cells through membrane is located at the surface tend to sense the environment, they detect their chemicals, and then they transfer the information to the interior. And depending on the nature of the information, an enzyme will be produced and will cause the cells to respond accordingly. We find that chemotaxis plays a vital role in reproduction, tissue repair, drug delivery, tumor invasion. It's involved in metastasis, atherosclerosis states of disease. It has really quite a number of different applications. Okay? More recently, work has been done on bacterial chemotaxis. It's one of the better documented systems in biology. In particular, traveling bands of bacteria have been observed when one introduces a population of cells of E. coli in a capillary tube accommodating oxygen and an energy source. And what we're looking at here is the chemotaxis movement of E. coli cells towards an aspartate. They're inoculated in a soft agar plate, which could observed chemically, and what we want to do now is look at how we can model that using So the system of equations that we're looking at here is these systems of partial differential equations. Okay? Here our dependent variables are n, j, and s. N and j are the macroscopic cell density and the flux respectively. S is the distribution of the signal, and that's what we're really going to be interested in. We have a proliferation of the host rate, which is a function of the distribution of the signal itself. We have the speed of the cells. We have the consumption rate, degradation of the cost, and so on and so forth. Okay? This is a particular system that we're looking at, which only has, the signal. We don't have equations governing the distribution of food. So this particular case is a case of starvation, where we have the low presence or the absence of nutrients. And so what happens here is that after giving the oxygen in the environment, the cells will secrete a particular signal, and that signal is then consumed by the other cells. Okay? So that is the way this particular system works. So if we analyze this particular system for symmetries, it's really quite messy. Okay? In the case of zero growth, we have the 6 symmetries given by 33 and 34. As you can see, they're quite messy. Okay. If you have constant growth, you also have 6 symmetries given by 35, 37, when we have a particular relationship between the constant, and 38 and 39 for another relationship between the constant. I should point out there's also an infinite dimensional symmetry linked to the linear equation that arrives at the system, but I haven't given that particular symmetry. Now, if you look at this horrible set of symmetries, it's difficult to see whether one can make. Well, it turns out that in each set of symmetries, you can always write down the variance. So you can always write down the new variables that you use for to substitute into your original system. Now, again, this looks really quite messy. And instead of putting these horrible expressions back into the original system, we then look at the biological implications of these different expressions and see if they make sense. Okay? It turns out that biologically we can remove a number of the extraneous parts of these particular transformations, and indeed the only transformation you need to worry about is this very simple system here. Now, this is the old adage of make your analysis as simple as possible, but no simpler. And the reason I mention this is that usually in mathematical biology, as I mentioned earlier, one would just substitute u equals x minus ct to find your traveling wave solutions. And that is the simplest thing you could do. However, it turns out that if you do that, you cannot define traveling wave solutions that are biologically relevant. In fact, what makes the difference is this exponential, which multiplies x. This constant C2 is an arbitrary constant that we put in by hand as a result of the symmetries of the system or linear combination of the symmetries, and this makes all the difference in analysis. If we didn't have that particular constant, we would not be able to find biologically relevant traveling waves. So substituting this system of covariance into our original system of TDEs will yield this system of ODEs. This system of ODEs looks a little nasty, but it is indeed tractable. In the case of zero growth, interestingly, non-diffusing biologically relevant traveling wave solutions do not exist. And this is really quite interesting, that you must have diffusion in the system to obtain traveling solutions that are relevant. So the diffusion actually stabilizes the system and prevents these solutions from blowing up. The solutions that we found are given here at 47 through to 49, where I are your Bessel functions. Okay, so you can write down the solutions explicitly. And as you can see here in this particular figure, this is the distribution of the cells on the left-hand side and the signal on the right-hand side. And you can see the obvious traveling wave nature of these particular solutions here. Okay. In the case of consistent growth, where h of s is now a constant, Again, you can find traveling wave solutions. I've just looked at the high chemotactic sensitivity case here. Okay, and again, we have solutions when the diffusion is non-zero. This again is plot of both the distribution of the cells and the signal over a particular time. Okay. For this system, just to give a summary of the results, I haven't presented all the results, but I thought the summary would be useful. And what you can see is that traveling wave solutions force us to have non-zero diffusion constants. Okay? In the case of zero diffusion constants, we have to relax some requirements of the behavior of solutions for you to train traveling wave solutions. But if you want traveling wave solutions in general without any diffusion, then you have to have diffusion in your particular. We thought was quite an interesting result. So those were just a few examples of applications of symmetry analysis to problems that arises in mathematical biology, both ODEs and PDEs. Across this area is really still quite open for lots of analysis. In particular, I've alluded to higher dimensional systems of first order ODEs. They can be quite complicated if you do them in general, but they arise in a number of interesting applications. We can look at applications in waterborne diseases, in particular, tick-borne diseases, where you have a vector and a host, which are really quite interesting. And so these systems are all to analysis. Okay. One area that we haven't really looked at in South Africa, but does bear a little bit more attention, is that of identifiability. And this is the idea of being able to determine the constants in your, or the parameters in your various models. There is a belief that one can do this from a dynamical, from a group theory, and that would be interesting to look at. But the other point about identifiability is it can also help restrict those very general systems that I presented to you earlier. And so in that sense, you can start honing in on biological relevant systems to look at the idea of identifiability. Since biology tends to want to mimic, mathematical biology tends to want to mimic real life, more perhaps than some physical systems, one tends to really use differential equations to get an accurate analysis of exactly how systems evolve. Unfortunately enough, much has been done with regards to the analysis of these differential equations from a symmetry perspective. I believe a few years ago, Faisal Mohammed and some of his students looked at symmetries of stochastic differential equations. I think it is well worth revisiting, given the proliferation of stochastic differential equations in mathematical biology. And finally, I just want to make a point for the further understanding or analysis of the first integrals or the conservation laws that arise in mathematical biology. We understand what all of these mean in mathematical physics, but it isn't clear to me that there is a full understanding of the implications of these first integrals in biological systems. And there are some colleagues down at Sathasiva who are working on this, and we'll be able to do a little bit of work on this in the coming weeks. Okay. Just remains for me to thank my students from throughout Africa who have worked on this project with me in various problems, to thank my funders, and finally I need to have a shout out to my chiropractor, Keshlan Walker, without whose efforts I would be able to be sitting here today giving you this talk. Okay? Thank you very much.
Yes, thank you very much.